So this is just showing that exact idea. Sperm cell, where N is 23. Egg cell, where N is 23. There would be 23 of these, 23 of these. All right. Once fertilization takes place, you've got the 23 that came from dad, and you've got the 23 that came from mom, which gives you 46, 2N. Now, homologous chromosomes is another word that we can use to describe them. All right, homologous chromosomes are the idea that they're similar in size, similar in shape. Each pair, all right, would be a homolog. These would be a homologous pair of chromosomes. They're similar in size, they're similar in shape, and they're similar in genetic content. By similar, what I mean is that perhaps that gene right there, all right, this is going to be dad's chromosome, while this is going to be mom's chromosome. These are the two chromosomes. Let's say you're number one, so you got one from mom, one from dad. Let's say that this is a gene for blue eyes, whereas this is a gene for brown eyes. They're similar in genetic content. It's still the eye color gene. It's just a different manifestation of the gene. So the idea that, like, it's still coding for the same type of trait, it's just exactly what the trait looks like that's different. Each homolog comes from mom, all right, or dad. So homologous pairs are referring to, you have 23 pairs of homologous chromosomes in all of your cells. 23 from mom, 23 from dad, giving you 23 pairs, and they're homologous because they should be similar to give you a full set of genetic information. You can also describe your chromosomes based on what they're coding for. You've got autosomes, all right, that don't have anything to do with gender. In our case, we've got 23 pairs, which would give us 44 total, right? They're not directly involved in determining gender, right? So we'd have 44 autosomes versus sex chromosomes. We've got one of the 23 pairs determines gender. This would be pair number 23, right? Only sex cells is pair number 23. It can be the X and the Y chromosomes. XX is female, XY is male. So, interesting thing. If each gamete that a parent produces can only carry one number 1, one number 2, one number 3, one number 4, etc., all the way down to one number 23, when it comes to mom's eggs, mom's egg cell can have an X, or mom's egg cell can have an X. However, dad's sperm cell can have an X, or his sperm cell can have a Y. So for the gender for the gene for maleness, or the chromosome for maleness, is actually passed on from fathers. Fathers' sperm cells are what determine gender. It's not something that's controlled, guys. So when we start joining, if this egg and this sperm join together, we've got a girl. If this egg and this sperm join together, we've got a girl. If this egg and this sperm, we have a boy. If it's this egg and this sperm, it's a boy again. It's a 50-50 shot. Every time people reproduce, it's a 50-50 chance for a boy or a girl. Never changes. Always just as likely to be a male as it is to be female. All right, then we have information about chromosomal abnormalities. Normal is 46. All right? 47 would mean that you have an extra. 45 would mean not enough. You're missing one, all right? 46 is a normal number. 47, 45, they are not. How can we tell if it's normal? Well, we can look at something called a karyotype. This picture right here, this is a karyotype. Based on this information, our two number ones, we've got two number ones, we've got two number threes, I'm sorry, twos, threes, fours, fives, and we keep going down, and we've got pairs of homologous chromosomes, right? Because one of them came from mom and one of them came from dad, right? We've got two number 19s, two number 20s, 21, 22, and if we look here, we've got one big one, and we know that the Y had to come from dad, so this is a male, right? This is a karyotype for a male. How about in this one? All right, we should be able to go through and we should be able to look. Two number ones, twos, threes, fours, fives, right? Two number sixes, sevens, eights, nines. And if you look, we've got similar banding patterns. That band, all right? Look at that indent and that indent, right? Pale band and a pale band, right? We've got similar banding patterns. They're paired up based on their size and their genetic information. If we keep going through, we should be able to see that everything is working out just fine until we get here. We've got three number 21s. Something's wrong. We got two number 22s, and we got two X's, which means we have a female. This means that in this case, the total number of chromosomes is 47. This is 47, and it's XX. So we know that it's female, and it has an extra chromosome. We don't know where, but we know that. Well, we do know where. We know it's on the 21st chromosome based on the karyotype. But based on just that 47, XX, we don't. Well, it tells us, though, that something is wrong. In this case, all right, we have an abnormality, and that particular abnormality is something that's called trisomy, all right? There are three bodies in the 21st position, all right? And by bodies, we're talking about chromosomes. Soma, remember, body. Trisomy, three bodies in the 21st position, three chromosomes in the 21st position. We typically call it Down syndrome. All right, so a person with Down syndrome has an extra chromosome. It is a genetic mutation, a genetic disorder. And we know that this is a female. The way that you get the information for a karyotype is a process called amniocentesis. All right, and amniocentesis is a way that the doctor is going to extract, all right, amniotic fluid. That's the fluid that the baby is floating in. If you look at our picture right here, we've got a big needle poking into the amniotic sac where the baby is. All right, that method is going to extract some fluid where the baby has been floating. Fluid typically has some of the cells from the baby that the baby has, rubbed off or whatever. And you're then able to get a cell sample. If you can get a cell sample, then you can get a DNA sample. And then if you can get a DNA sample, obviously you have a picture of the chromosomes. All right, you're going to be able to do a karyotype. All right, you're going to be able to look at this particular karyotype and say, is everything okay with the baby? Right? This is something that you would do, obviously, before the baby is born. Right? So, amniocentesis is what you use to create a karyotype. Problem is that there are risks. You're sticking a needle into the abdomen, right, into the uterus, right, into the amniotic sac. You're piercing the amniotic sac with a very thin needle in order to extract that fluid. Right? There are dangers to the unborn baby. In addition, what if there is something wrong? What decisions do you have to make? What do you have to deal with? All right? Is it okay for things to be wrong? Are there dangers if there's something wrong? All right? What if your child is going to be born with Down syndrome? Is that something that you can handle? All right? So amniocentesis is something that, is useful information, but also can raise some interesting questions for a parent to have to make. So this wraps it up in terms of our genes and our chromosomes benchmark.